Hello, everybody. Welcome back to another wonderful biology class. Today, we're going to be looking at complex patterns of inheritance. And the big focus question for today is going to be, what are examples of complex inheritance? It's a good question, so let's get into it. So the first pattern we're going to look at is incomplete dominance. Then we'll look at codominance. Then we'll look at multiple allele inheritance, or multiple alleles, and then epistasis, and then sex-linked traits, and then polygenic traits. All great stuff, all good to know, so let's go. Review vocabulary that you should hopefully know is a gamete, which is going to be a sperm cell or an egg cell, a spermatozoa or a oocyte that is involved in sexual reproduction, which goes from 1N as a sperm or an egg to a 2N diploid organism. All right, so let's begin with incomplete dominance. Incomplete dominance is kind of what it sounds like. It's where you have a dominant allele and a recessive allele, but the dominant allele isn't fully dominant. Okay, and so if I took two heterozygotes and I crossed them, I did a dihybrid cross, I would get a 1 to 2 to 1 ratio where 1 or 25% roughly, is going to be the dominant color. Okay, in flowers, it's oftentimes red, for example. And in 25% of the case, I will get the homozygous recessive color, which is white, for example. But in 50%, I would get a color that is roughly pink because the red is being expressed, but not completely. So the pigment is still being produced, but not enough of that pigment is being produced to completely color it red, so it winds up pink, which is a mix of red and white. And so if we look at our carrots here, for example, you might think of your gray carrot here as a very recessive trait, and your lovely red-orange carrot here as the dominant trait. And here in the middle, you have kind of a reddish, grayish carrot that's somewhere in the middle. That would be incomplete dominance. Now, moving on to codominance. Codominance is where both traits are expressed in a, well, almost 50-50 manner, we'll say, right? So a good example of this is sickle cell disease. In sickle cell disease, you produce a crescent moon-shaped cell called a sickle cell when you have the recessive disease, or one of the diseased alleles. Okay, not recessive, it's codominant. So when you have the diseased allele, your body produces damaged sickle cells. Now, when you have two of these dominant alleles, all of the cells are sickle cells. And this causes what's called sickle cell anemia, full-blown sickle cell anemia. And oftentimes, this is a fatal disease. It causes blood clotting in the capillaries, so you get a lot of random bruising, and it can also cause blood clots in the brain, which lead to strokes and eventually death. So it's very bad to have a homozygous sickle cell trait. However, in sub-Saharan Africa, where there's a mosquito-borne parasite known as malaria, having the sickle cell trait partially is actually kind of a good thing. You see, normally the malarial parasite will dig straight into a healthy red blood cell and eat it, and then of course multiply and make more malarial parasites and give you what's called malaria, whose symptoms include things like high fever, vomiting, diarrhea, nausea, abdominal pain, and in young and elderly individuals, oftentimes, very sadly, death because of dehydration. Or apoxia, which is low blood oxygen levels because so many red blood cells have been damaged. Now, for a person who has one healthy red blood cell trait and one sickle cell trait, both normal healthy red blood cells and sickle cells are produced at the same time in a roughly 50-50% ratio. Well, what does this mean? It means that when a malarial parasite enters that person, it can attach to the healthy red blood cells and multiply. But if it attaches to a sickle cell, Because of the odd shape of the sickle cell, the malarial parasite is not able to eat its way into the cell, and so it gets stuck. And eventually that gets filtered out of your blood and killed by white blood cells, and then you excrete the waste as urine. And so having one of the sickle cell trait, this codominant trait, is actually advantageous if you live in a place where malaria is common. Now, this is not a polygenic trait. This is multiple allele trait. Sorry, thank you. In multiple allele traits, you have more than a dominant and a recessive allele. That seems pretty straightforward. This just means that instead of having a dominant red or recessive white for flower colors, you can have some other allele entirely different that is a possibility. And a great example of this is blood groups or blood types. Okay, in humans, there are 3 different blood types. There's type A, type B, and type O blood. And these are 3 different alleles. There's an O allele, a B allele, and an A allele. And what they do is they code for the presence of an A receptor, a B receptor, or no receptors at all on the surface of your red blood cells. Okay, and so what does this mean? It means that you can have several different types of blood. You can have type A blood, which is when you have 2 A alleles, or an AO allele, because the O does not produce any receptors. So you can get type A from having AA alleles or AO alleles. You can get type B blood from having BB alleles or BO alleles. And you can only get type O when you have OO alleles. But you can also get another type, which is type AB, because you have one A allele and one B allele. And this is interesting when we talk about determining whose child someone is. Blood typing is actually one way that this is oftentimes figured out. So that's multiple allele inheritance. Another multiple allele type of inheritance is involved in fur color, in rabbits and in other types of animals, where you have one allele for white fur, one allele for tan brownish fur, and one allele for completely jet black fur. And you can get a nice variety all the way from snow bunny white to the darkest forest black bunny, based on the combination of these alleles. Now, epistasis is an interesting form of inheritance because this happens when you have one type of dominant allele completely cover up another type. Okay, so allele A produces some kind of phenotype that completely masks or hides the phenotype of the type B allele. So what would be an example of this? Well, perhaps you have the allele for black fur and the allele for tan fur. Well, in a situation where these 2 alleles were epistatic, or they expressed epistasis, the fur would be black, even though the pigments for brown were also being produced. You just can't see them. They're there, but they don't show up in the phenotype because the phenotype looks like the dominant phenotype. Now, looking at other types of inheritance, you can have what's known as dosage compensation. And for the X chromosome carries a variety of different types of developmental genes for both females and males. The Y chromosome, on the other hand, mainly has genes that relate to the development of male individuals. Now, in females, one X chromosome is inactivated in the cells, and so therefore the other one is thereby expressed. Okay, and so your inactivated chromosome becomes what's known as a bar body. And that inactivated chromosome is just the chromosome wrapped around histones very, very tightly so that none of those X genes can be expressed. And so this is known as dosage compensation or X inactivation, which results in one of the X chromosomes not being expressed at all, and occurs in all mammals, including us humans. So oftentimes, the healthier of the two X chromosomes is the one that's expressed, and the less healthy X chromosome is entirely inactivated. Now, does this always happen? No, there's some disorders in which this doesn't happen, and it results in some developmental issues and oftentimes the need for hormone treatment. But a healthy individual, this is the way that it works. Now, chromosome inactivation is oftentimes observed in things like coat colors in cats. And you can see this because you have areas of the cat that are one color, or would have been one color, but because of chromosomal inactivation, the color is not produced in that area. Okay? And so a lot of the fur color in cats, for example, is a result of the inactivation of one of the X chromosomes during development. And again, that brings us back to the idea of Barr bodies, which are inactivated X chromosomes, and they can be observed in cells directly with a light microscope. They tend to be darkly stained, and they don't look like an X chromosome. They just look like a linear line of hard nuclear material. All right, that's enough for this lecture. We're going to have another one. It's kind of a lengthy one. Sorry about that. But thank you so much for listening. I've got some questions for you, so go get them.